Two scientists embark on a voyage across the sea of biochemistry in search of one protein so dear to their hearts.
I have to find my Myo. I have to tell him how old sea turtles are. That heme-containing muscle protein, myoglobin, means more to me than all the fish in the sea. Fish are frozen, not food.
Liquid nitrogen keeps the cells cold enough that enzymes don't degrade our precious mayo.
She means Myo. Gotta break open those cells keeping us from Myo and suspend them in extraction buffer. Just keep shaking, just keep shaking, just keep shaking, shaking, shaking.
Mino.
You mean Myo.
Myo will be swimming around in the supernatant after centrifugation, happily protected from oxidation eels, serine, protease, sharks, and enzymes.
Now that we've isolated our dear Myo, let's just salt him out of solution, enriching the concentration.
I shall call him Salty, and he shall be mine, and he shall be my Salty.
If we gradually increase the concentration of ammonium sulfate and centrifuge in between, eventually Myo will precipitate out of solution.
High salt concentrations, even higher than in our lovely ocean home, disrupt water-hydrogen bonds, so there is less of an entropy benefit from the folding due to hydrophobic interactions. Proteins precipitate, no longer in their native states.
At first, we may not see Myo, but he'll crash out of solution and back to us. The least soluble proteins will precipitate first. Myo will be surrounded by all the other proteins in the crude myoglobin extract, so let's use size exclusion gel chromatography to find him.
We'll use a hollow column of packing material which has different shapes and sizes of porous beads.
The larger proteins will diffuse faster through the chamber than smaller proteins because the porous beads slow the smaller molecules down, whereas the larger beads move unhindered.
We use a similar solution to the extraction buffer described earlier that will keep my precious Myo safe.
Right, we just have to make sure that after we load the crude extract in that column has enough flow so that it doesn't run dry, or else all of our work will be lost in the abyss.
You're very right. So we'll let the column run and collect 1 ml of eluate in 50 tubes, and we'll test a range of tubes for the presence of Myo.
Let's test absorbance of different samples using the nanodrop to first find proteins around 280 nanometers, and then look for a heme group at 415. We found him!
Hello, fellow scientist dudes! Did anyone order a package from P. Sherman 42, Wallaby Way, Sydney?
P. Sherman 42 Wallaby Way, Sydney! Nope, not familiar.
Dr. Dory, this package has all the supplies for gel electrophoresis, our last step in finding Myo.
Didn't we already find Myo?
We need a way to make sure it's really him. When we run a charge through this gel, negatively charged proteins travel a certain distance towards the positive charged end based on size.
Dude! SDS is a sweet detergent for retaining primary structure while making proteins negatively charged.
Once we load the samples into the individual wells of the gel, it'll run for 1 hour. How far the proteins get will be based on their sizes, with the smallest traveling the furthest. This gel is clear. How are we going to identify which one is Myo?
Don't worry, little dude. You just need to use Kumasi Brilliant Blue dye in order to stain the proteins on the gel. The bands will appear in different places on the gel, and you can compare those with where you know Myo's band would be, at about 17,699 Daltons.
It's him! It's really him! You can see tons of other proteins in the first two lanes from the first two fractionations at 30% and 45% saturation. They must have come out of solution at those concentrations of ammonium sulfate.
And you can see specifically myoglobin bands in lanes 4 and 7. Lane 4 was our third fractionation, so myoglobin must have precipitated most at 60% ammonium sulfate. Lane 7 was one of the two size exclusion fractions that showed the most protein on the nanodrop. We found Myo!
This film brought to you by the Center for Scientists' Hand-Size Diversity.